Welcome as our opening keynote lecturer, Professor Sir Michael Barrage. I'm sure that Mike Barrage's name will be known to many of us as the discoverer of the inositol trisphosphate as a second messenger, one of the pioneers of calcium signaling. He became a fellow of Trinity College here in 1972 and then was elected.
Trinity College, Cambridge University. It was during his years here as a fellow that Sir Michael Barrage made his groundbreaking discovery of a universal second messenger in cells.
I find Trinity College to be tremendously inspirational. I walk over from the laboratory. I come into this wonderful Elizabethan dining hall every day for lunch. Here we have a portrait of J.J. Thompson, one of the great physicists in Trinity, and he was noted for the discovery of the electron. Lord Rutherford noted for his discovery of the nucleus.
Rutherford and Thompson laid the foundation of atomic physics.
And over here we have one of my great heroes, and this is Lord Adrian.
He discovered the way neurons interact with each other.
Here we have two other very famous physiologists. One is Sir Alan Hodgkin, and the other is Sir Andrew Huxley. They worked out the ionic basis of the action potential that Lord Adrian had discovered. So in a way they complete a circle. And seeing the portraits of all these extraordinary scientists does provide a lot of inspiration, and it sort of makes me really want to carry on and do exceptional research.
Barrage's PhD studies was in insect physiology, here at the Department of Zoology at Cambridge University. In one later research using blowflies, Barrage dropped solutions similar to insect blood onto the cells of their salivary glands. Hormones in the blood can't penetrate cells. They bind to receptors and transducers on the cell membrane and stimulate the cells to secrete fluid.
Just when you're going to have a nice meal and your saliva starts flowing, that's exactly what happens in the fly. So I was trying to study what goes on inside the cell to activate this secretory process. We knew that there was something between the membrane and the secretory process, and there was a big question mark.
An unknown messenger. For about 12 years, Barrage worked daily in the lab between tutoring students at Trinity College, probing cells which are so small 100 of the minuscule matter can fit on top of a sewing pin.
It was very much like looking for a needle in a haystack, because there are lots of molecules in the cell, and somehow one had to find this unique second messenger.
He knew that for a messenger to work, it had to be produced rapidly.
And I was able to isolate this messenger and measure its formation in time, and I found this very rapid increase in IP3.
To make sure the chemical is really IP3 or inositol triphosphate, Barrage had to obtain sample molecules which were rare in the early 1980s. He says he was very lucky to find them at the Barrage Institute in Cambridge.
So if we artificially introduced the messenger into the cells, it could activate the secretory process. And that's how we knew that IP3 was in fact the missing link in hormone action.
His paper on this discovery was published in late 1983.
Not bad.
It's just like doing science. Occasionally about 10% of the experiments work, and it's like golf with me. About 10% work quite well, and the rest are abject failure. Like 2 in the bunker here, and several down at the bottom of the green. There aren't many eureka moments in science, but I can remember very clearly when I did a definitive experiment, and it clearly showed that IP3 was doing something very important. I did have a moment when I suddenly realized that this might be the answer. Good boy. Even my wife remembers me coming home that night.
And said, I've discovered something really important. He actually said that. He was so excited. Oh, I was thrilled, of course, because I knew he was working so hard. I knew he had the necessary mind to ask the right questions. Also, I'd done some research for him at one stage, and it was so boring that I gave up. And he had to complete the project.
Doing research is actually quite a lonely job. You spend a lot of time by yourself reading and doing the experiments, and the whole responsibility for bringing up the family was on Sue's shoulders.
At the time, Sue was working full-time as a midwife. The couple had met at a dance when they were both students in Cambridge. They married in the United States, where Michael did some postdoctoral work. Their daughter and son were born there. Now, both Michael and Sue are retired.
But I'm still working, perhaps far too hard, but it's in my blood, really.
Barrage still does research at the Barrage Institute, which he joined in 1990.
Hi, Martin.
Hi, Mike.
Okay.
Yeah, good.
Dr. Martin Goodman was Mike's PhD student at Cambridge.
He just has this colossal grasp of so many different areas of science that it's a wonderful resource to have. He's a walking library. And the social interactions, of course. He's a very generous individual with his time, with his knowledge.
Barrage's research had shown in the cell membrane, each molecule has two fatty tails and an inositol phosphate group. When a hormone attaches to a receptor on the membrane, it breaks the link, leaving the fatty tails in the membrane and liberating the inositol triphosphate, the second messenger, which then goes into the cell.
The IP3 functions to produce calcium, and the calcium then activates different cellular processes. It can control, for example, cell growth. And there are some indications that this signaling pathway, if it goes wrong, can cause certain diseases. And for example, certain types of cancer, particularly leukemias, seem to result if this system is overactive. So it gives too much cell growth stimulation, and therefore you get excessive growth.
And it's now known why the drug lithium can control manic depressive illness.
We found that it actually interferes and inhibits this signaling pathway. Excessive signaling within the nervous system may lead to various mental diseases like manic depressive illness.
Recently, scientists also found that when a sperm binds to an egg, it injects a transducer and the egg begins to make IP3 and generates every 2 minutes pulses of calcium, which activates its development.
So now that we understand how those pulses are produced, we may be able to improve the process of in vitro fertilization.
There are also new insights into heart disease. These are cardiac cells artificially stimulated with calcium.
Each time this goes red, there's an elevation of calcium. Calcium is causing these cardiac cells to contract.
Normal cells should have uniform elevation of calcium and uniform contractions. But bad diet, excess weight, hypertension, or high blood pressure make the heart pump harder and activate new genes.
One of the genes that are activated increases the IP3 system in an attempt to try and get the heart to work better. But it also starts to produce these arrhythmias, and that's what leads to sudden heart death. For a long time, there was absolutely no understanding as to the cause of these arrhythmias. So there's a lot of interest now in trying to develop ways of regulating the IP3 through the application of drugs to try and control the generation of these arrhythmias. I decided to retire to the countryside, and we live in this converted watermill. And it's a magical place to live in, and I feel I've got my own water hole here. It reminds me a lot of my home in Africa.
Sir Michael Barrage was born in Gatuma, Rhodesia.
Well, as a young boy, I was surrounded by the African bush, and I was always very interested in the wild animals. I had a wonderful biology mistress, Pamela Bates, and she was the one that really got me interested in studying biology. She was very influential in my whole career.
Studying for his Bachelor of Science at the University of Rhodesia, Barrage was inspired by another teacher to pursue his PhD at Cambridge under Sir Vincent Wigglesworth.
The Albert Lasker is quite an important award from America, the King Faisal Prize from Saudi Arabia.
His discovery of IP3 has garnered a host of awards, the latest being Hong Kong's Shaw Prize.
One of the nice things about prizes is that one goes to lots of different parts of the world to receive these awards, so there's quite a sort of international array.
His major project now is to complete a book, the first ever to be published on the web with no hard copies.
Everybody says to me, "Oh, but the internet is so full of information." And my answer to that is, yes, there's lots of information on the internet, but there's not much knowledge. And by knowledge, I mean this business of being able to show how things are interrelated and bring the whole system together to integrate all the information.
One day, Sir Michael Barrage will return to the hallowed halls of Trinity College. Sir Michael, as a fellow of Trinity College, your epitaph will one day be here in this chapel. What would you like it to say?
I think what I would like is for it to say, "A life spent in the pursuit of excellence of science, assisted by a loving family." And I think that would encapsulate my life as a scientist.